Hello and welcome to Positron Academy. This video will be talking about DNA damage response. Starting with the time scale of the effects of exposure to radiation on any biological system. This time scale is basically divided into three phases, namely physical, chemical and biological. So starting with the physical phase is if we take into account a high-speed electron, it usually takes about 10 to the power minus 18 seconds to pass through a DNA molecule and about 10 to the power minus 14 seconds to pass through an entire mammalian cell. So as it does so, it interacts mainly with the orbital electrons of these atoms, and via its interaction it causes excitation or ionization of these orbital electrons. And if a sufficiently energetic electron is created, then this secondary electron then passes through its neighboring atoms and leading to a cascade of reactions. So this damaged atoms and molecules are giving rise to the free radicals, which involve in reactive reactions. And as we know that excitation and ionization are leading to these chemical bond breakages, so the free radical reactions which are taking place is actually a competition between the scavenging reactions which want to eliminate or neutralize these free radicals compared to the fixation reactions, which are going to be making the chemical change permanent, which is fixating the chemical change on these biological systems. So this was the chemical phase, which deals with the process of how these damaged atoms and molecules are reacting with the biological components. Moving forward to the biological phase, it deals with the enzyme reactions mainly, which are going to work against these damages, and also along with that are the repair processes which are going to repair any lesions caused to these molecule, especially the DNA. And we see that the cells which have undergone these which have these defective DNA, they usually die after a few divisions, a few mitotic divisions in the line, where a defective genome leads to their instability and loss or death of the daughter cells. So another example of this cell killing is called as compensatory cell proliferation. So this occurs both in normal tissues as well as in the tumor cells. Moving forward to early effects, it is seen primarily when stem cells are killed or a loss of the stem cells take place, and the examples are seen as the breakdown of the mucosal layer as well as hemopoietic damage, and slowly the late effects start to appear, which are like examples are fibrosis as well as telangiectasia of the skin. So on a more later time scale, we see the appearance of secondary tumors, and this is named as radiation carcinogenesis. Moving forward to DNA being the principal target, here are the three experiments which had provided evidence for DNA being principally the target for cell killing. It's because if you consider irradiation, ionizing irradiation, it will be depositing its energy randomly on all the cellular organelles. But if we see mRNA proteins and other molecules, then these are abundantly present in number, as well as their turnover is high compared to DNA, which is present in only 2 copies, and its turnover is very less, so making the DNA more susceptible to damage. And as we know that since the DNA is forming the biggest target in the cell in the form of the nucleus, as well as it being the central functioning location for the entire cellular survival. So any damage to DNA will be lethal for the cell. Coming to experiment 1, which dealt with small polonium needles which radiated short-range alpha particles. When these were injected into plasma membrane or the cytoplasm, the cell could handle even high doses of radiation, and the cell was surviving. But even 1 or 2 alpha particles radiating the nucleus, it was leading to cell death of imminent cell death. So this also proves that the nucleus is the target. And coming to experiment 2, where the plasma membrane was labeled with iodine-125 concanavalin, and the DNA was labeled with tritium labeled thymidine. So these radiations to these radioactive these radiations were compared with the homogeneous cell irradiation with X-rays, and we see that there is a direct probability that if the more the dose to the nucleus, the higher the chances that the cell will die. So this also proves that the nucleus is being the main target during irradiation-induced cell death. Coming to experiment 3, where Chinese hamster ovarian cells were taken, and all the cells were synchronized in the G1 phase, with the plasma membrane being radioactively labeled with iodine-125 concanavalin, and nucleus was labeled with iodine-125 iododeoxyuridine. The delay in the cell division was checked. So as we know that radiation-induced damage will of course influence the cell division. So what we see is that the batch which was the batch of cells which were radioactive, where the nucleus was radioactively labeled, was showing highest division delay compared to the batch of cells where the plasma membrane was radioactively labeled. So even this proves to us that the nucleus is the most radiosensitive target for irradiation induced cell killing. Coming to DNA damage sensors, so when a DNA is damaged, that site of damage is sensed by these different protein complexes, and this site of damage is referred to as the IRIF, which is ionizing radiation induced foci. So as we know that phosphorylation leads to activation of any signaling. So phosphorylation is mediated through 3 related classes of kinase proteins, which are the ATM-MRN complex, the DNA dependent protein kinases, and the ATR-ATRIP complex. Coming to the MRN complex, which consists of 3 proteins, namely MRE11, the RAD50 protein, and the NBS protein. So explaining the MRE11, which is called as meiotic recombination. It is an exonucleus with an ability to unwind DNA locally. So what it does, it attaches the phosphoesterase, which is the nuclease domain. It binds with the 3' strand of the broken DNA. And this MRE11, it helps in binding of NBS1 and RAD50, which usually directly do not interact with each other. So RAD50 is a V-shaped protein with a zinc hook domain, and this allows the RAD50 molecules to dimerize and then attach the DNA ends together. So this is the zinc hook domain, and the MRE11, two MRE11 proteins bind to two RAD50 molecules. And finally the MBS protein, which is the Nijmegen breakage syndrome. It is responsible for activating the ATM-ATR protein. So the NBS1 protein then attaches to the MRE11, then attaches to NBS1 protein. So altogether, the MRN complex consists of two MRE11 proteins, complex with two RAD50 proteins and one NBS protein. Double-strand break induction activates poly-ADP ribosylation polymerases, which is called as PARP1, and it attaches the ADP ribose units to the chromatin-bound proteins. So this damaged site also recruits the MRN complex, which binds to the double-strand breaks, and this MRN complex will subsequently dimerize and also bring forth the ATM molecules. So here we can see that the ATM dimer, which was inactive, has been activated by the NBS1 protein. So what happens? The MRN complex is sitting at the end of the DNA double-strand break, and the rigid arms have opened up to more flexible arms, which have caused the dimerization of two MRN complexes together. And the ATM dimer then sits on the NBS1 protein and is phosphorylated to form ATM active monomers. And further, what happens? The cascade, the signaling cascade is activated by these ATM monomers. So this results, this monomerization of the ATM protein further leads to phosphorylation of the H2AX proteins, which are around the broken chromatin, and this H2AX phosphorylation leads to the recruiting of repair proteins, which are BRCA1 or CTLP, to the site of damage, and together this complex of DNA repair proteins along with the ATM and MRN complex, they bring about the repair of the damaged DNA. This is illustrating the change that occurs when MRN complex, which was previously unbound, when it tethers to the DNA ends, it is in bound state, and then it opens up and it leads to dimerization via the zinc hooks, and that's when the inactive ATM sits on the NBS1 protein and gets phosphorylated to form the active monomers, and the signaling is continued. So here we can see the V-shaped RAD50, which is then opened up to form a dimer of the MRN complex with the ATM dimer also activated. And this is the overall structure of how ATM and MRN complex together bound to the DNA double-strand break brings about the repair. Coming to the ATR-ATRIP complex, first let us see how the damage is sensed. That is done by the sensing the presence of single-strand DNA gaps, unlike for ATM, which requires the presence of double-strand breaks. So ATR can sense a wide range of damage. So one of them being the presence of cis-DNA gaps, which can be caused by replication stress or even damage due to irradiation, which stalls the replication fork, and even by resected double-strand breaks, which leave behind as a gap of single-strand DNA. So this cis-DNA ds-DNA junction is what recruits the RPA. So RPA is replication protein A, and what it does is it covers the entire single strand DNA, and a chain of 75 units is what brings forward the recruitment of the ATR as well as the ATRIP. So ATR is actually the full form. The full form of ATR is ATM and RAD3 like protein. It is a kinase. It has kinase activity, and ATRIP is ATR interacting protein. So both ATR and ATRIP is activated by top BP1, which is DNA 2' 2 binding protein 1, and via its ATR activation domain, it phosphorylates ATR and ATRIP. So what ATRIP does is that it helps in localizing ATR and to the site of damage, as well as it initiates the interaction of RPA with ATR. So now that ATR, ATRIP and RPA are in place, let's look into RAD17 complex. So RAD17 is an replication complex like RFC like protein, which is part of the replication complex, and RAD17 along with 4 subunits of RFC forms the RAD17 complex, and that is also brought to the site of damage, and it adheres to the RPA. So the RAD17 is one of the proteins along with RAD9, which is responsible for recruiting top BP1 to the site of damage. So what happens? Top BP1, it phosphorylates this 911 complex, which is formed by RAD9 and HUS1 and RAD1. So altogether these are responsible for, especially RAD9 is what responsible for bringing top BP1 to the site of damage. And top BP1, it phosphorylates the histone H2B of the chromatin at the site, and that is what helps in positioning of the entire complex at the site, and this leads to the formation of the entire complex, and ATR finally it then goes forward with its substrate activation, which begins with phosphorylating the CHK1, which is the checkpoint which is responsible for DNA damage checkpoint activation. So what this checkpoint kinase 1 CHK1 does is that it mediates the DNA damage checkpoint pathway via claspin, and along with claspin during the S phase, it is crucial for the stability of the DNA replication fork, and that is how ATR, along with it phosphorylating other substrates like P53, it brings forth the stabilization of the DNA replication and brings forth the repair pathways. So what is interesting to note is that unlike ATM, ATR is crucial for stabilization of the genome in a cell. So it is also crucial for the cell's survival as a result. So any mutation to this ATR protein is going to lead to downstream signaling which are faulty. So recent clinical trials are coming into effect, which are dealing with ATR inhibitors to be the cancer drug target. Coming to the signaling transducers, we have 5 transducers, namely ATM, H2AX, DNA, protein kinase catalytic subunits, along with ATR, ATRIP, and PARPs. So ATM and ATR, ATRIP I have explained in my previous part of the video. So H2AX is actually the variant of the histone H2A, and when it is phosphorylated, it is termed as gamma H2AX. So what is important is that it gives access to other the DNA repair proteins to the site of damage. Coming to the DNA dependent protein kinase catalytic subunits, they are actually kinases which are specifically they respond to DNA damage, in particular to double strand breaks, and I will be elaborating on their functioning and how it is carried out by the Ku70 Ku80 complex when I will be talking about double strand break repair by non-homologous end joining. So coming to PARPs, which are poly ADP ribosylation polymerases, I had mentioned about PARP1. So what PARP1 and PARP2 they are actually involved in the recognition and signaling of radiation induced DNA damage, and such as single strand breaks, any DNA nicks, and even of DNA double strand breaks. So their activation as a whole leads to the modification of many other downstream cellular proteins which are part of the signaling. So that can be like phosphorylation and even ubiquitination as well as sumylation, acetylation, and poly-ADP ribosylation. In the next part of the video, I will be talking about the different effector pathways. So stay tuned.